Good evening, everyone, and welcome to week 3. Now, first of all, a big thank you because in last test, say later on, because I got the mail, and yes, it was very nice on some of the students that they very humbly said that yes, there were two mistakes in the test. So I'm sorry for that. I think while preparing, I don't know why it happened, but I'll take care next time. Right? So yes, that was my mistake. Yeah, at two places, it should not have occurred, but I'm sorry. Okay, so let's start with the biochemistry, right? Now this week, say again the same style. On Saturday, we'll be having the test. Now, biochemistry is something where we have to remember a lot. So I try to simplify the things by certain phrases because they are important, right? So that's how we shall proceed. And as this is the topic for today, I mean, as I wrote that for today's topic, we'll be talking about fundamentals of carbohydrates, proteins, and the lipids and the fats. Now, what I request all of you is once this session is complete, go through it thoroughly. Right? I'll be putting the notes immediately, but go through it thoroughly because for next 4 days we won't be able to come back to this particular topic, and we'll be using this foundation, what we learned today, to learn rest of the topics. So these topics will not come again, right? And straight away over here, when it will be written like say omega 3, omega 6, 9, uh, all those saccharides, right? Sugars, straight away chylomicrons, straight away we'll be using those words. Where today we'll be going into the detail of that. Yeah, books part, not today because it is lots of ground to cover, right? Not today, but in this week, definitely, friends, will be going for the books part, right? Not today, but at least in a day or two, we'll definitely go for it. Okay, so let's start with our first part, that is the carbohydrates. Now, see, in carbohydrates, all these things they are related very much with the specific diseases. So when we talk about carbohydrates, it is like we have to say it like carbohydrate and sugars, right? Carbohydrate and sugars, because what carbs are, right? What carbs are, if we divide it into two parts, simple sugar and the complex sugar, right? And the complex sugar. And when we say that, what exactly the sugar is, nothing much. Just we remember that this is the most basic of it, that it has got carbon, oxygen, and the hydrogen. This is simple sugar. So when we have just one molecule of it, or we have got two molecule of it, we call it sugar. But when they are connected all the way, right? When they are connected all the way, so like this, right? All the way, when there is a long chain, so we call this thing as a complex sugar, right? So this is a big, long chain. So that's what is simple and the complex sugar. Now, will it make a difference? Yes, it will make a big difference, right? But before we proceed for that, sugar, now we are adding one by one the words. Sugar will now onwards it will call as the saccharides, right? Saccharides. Now there are many types of saccharides. So if there is one sugar molecule, right? So it makes sense to tell it mono, right? Monosaccharides. Now onwards we'll write just S for saccharides, right? To save time. If there are two, yes, you must have guessed, we'll call it disaccharide. If there are few, right? When there are few. So that means when we say few, it means there has to be a number. It is like say 3 to 9, right? 3 to 9. So they are called as oligosaccharide, right? Whenever the word oligo comes, oligo means there are few. So it is oligosaccharide. And then obviously when there are more than or equal to 10, so they are polysaccharide. So it means from that simple sugar, right, we now went into all the way to define monosaccharide, disaccharide, oligosaccharide, and polysaccharide. Our main interest is this monosaccharide, because monosaccharide is the currency. It is the currency which everyone uses, right? This is a currency which can go everywhere. It can cross blood-brain barrier, it can go to tissues, and that is what will be used for energy. So this is what? This is glucose. That's the glucose. This is what will be used, right? It will be the major source of the, sorry, it is the major source of the energy. Now when we talk about monosaccharide, right, let's go into bit more detail. Now keep orienting yourself properly. At any point of time, if it happens that you are drifting, immediately we'll go back, and I'll also keep on going back and forth so that you know that we are properly oriented, right? Okay. And it might happen that we'll be understanding it in such a way that at the end, today we'll try to go for the complete revision all the way, right? Because this part is very important, so that's why at very high pace we'll go for this revision, right, at the end. Okay, so now we talk about the monosaccharide. So in monosaccharide, one is known to us, right? What we talked about, glucose. In fact, it is the glucose only who will be playing the major role. But then there is one more. It is called as the fructose, right? Something which is in fruits, right? It will be having its own importance. But then there is third one, which is galactose, right? Galactose, lactose, that means we are dealing with the milk, right? So this is like a milk sugar, this is like a fruit sugar, right? And this is like a common sugar which we'll be using. Now these are all monosaccharides. When we'll combine two monosaccharide, right? So obviously it will become what? It will become disaccharide, right? So same way, say over here, let's make the disaccharide out of it. So when we combine, let's say, when we combine, let's say glucose and the galactose, now what we get is lactose. So this lactose, obviously it has got now two sugar molecules, so it would be disaccharide, right? Disaccharide. This is what is present into the milk, even in the say human breast milk, this is present, right? Let's finish this over here only. It will become much easier. So then let's combine fructose and glucose, right? So let me change the color. So we combine fructose and glucose. That leads to sucrose, right? So this leads to sucrose. Sugarcane, S for S, sugarcane, right? Very, very good quality of sugar. And finally, say when two, let's use the color. Okay, no problem. Let's use white only. So when this glucose and glucose, so this is glucose plus glucose. It means when the two molecules of glucose, so that leads to maltose, right? That leads to maltose. And this is what is used to ferment beer, etc. Right? Yeah, G plus G is maltose, so that is for the fermentation. So thus we have come now to monosaccharide and the disaccharide part, right? These are simple sugars, because when we said simple, that means either one sugar molecule or two sugar molecule. So the next would be what? It would be the complex sugar, right? So that's the next part, which is like complex sugar. In complex sugar, there are oligosaccharide, right? In which we said oligo means few, and the polysaccharide, that means many. Oligo means few. This is this oligosaccharide. It is galacto-oligsaccharide, right? Something which you get in that soyabeans. So galacto oligo, and then for saccharide we are just writing S, right? So this is what we get in soyabeans. Now these are short chains, relatively short chains, right? And galacto itself tells that this is the short chains of galactose, right? Of galactose. And then we have long chains. That's where the polysaccharide will come into picture. These are large chains, huge chains. And this is now called as, one more word we are adding, starch. Starch, right? This means what? This means because it's a long chain, right? It's a long chain. So over here, we will not draw the full chain, right? We'll just draw, and when I write S, that means sugar. So just I'll draw, and then we do it like this. It means it's a long chain. The bond which is present over here, can it be broken or it cannot be broken? That is what will decide that what exactly are we talking about. When we are talking about starch, the starch means what? It like rice. Rice, when we eat, we can digest. So it means this particular bond, right? This particular bond, it breaks. It can be broken. Our intestinal enzymes, right, can break it. It means it is digestible. Right? We can digest it. It will be broken down into smaller components, and then it will be absorbed, and all those processes we'll see in a short time from now. But yes, it can break. So that's the point, that if it can break, we call it starch, right? So all those like rice and potato, and but yes, they raise the sugar. Important point is actually, if you see the sugar, when you put on your tongue, right, you get a taste. When we put that rice or potato or wheat, we, though it is like such a high sugar, but no, we don't get the taste, right? It's not so that they are not sweet. It is because that they don't activate the taste buds because of those long chains, right? Their absorption for the taste bud, that is relatively very less. So that's the reason that they never taste as sweet, right? So we'll write no sweet taste, right? No sweet, no sweet taste, right? It was no sweet taste. Now, in case if this bond is so tight that if it cannot be broken, right, it happens that there would be certain situations and certain components in which this bond is not broken, right? Bond not broken by our any of the enzymes. Or even if it is broken, it is in very small quantity. So we'll just keep it not broken. If it is not broken, friends, then we call this thing as fibers, right? These are the fibers. So at times when it is said that for a diabetic patient, increase the amount of fibers. Now these fibers, someone cannot eat grass, right? So that's why these are like dietary fibers, right? Dietary fibers. So dietary fibers, they cannot be digested. Reason behind it is, yes, they are sugar, but the bond itself is so strong that our enzymes are incapable of breaking it. So therefore, they cannot be digested, right? If they cannot be digested, they cannot be broken, right? Delta pectoral triangle, again, right now we won't be entering into the anatomy part, right? We'll do one thing. I'm taking a small break just to, because this question I got it in mail also. And see, we'll keep one session that would be like a miscellaneous session, right? So whatever the doubt, say for example that delta pectoral triangle, no problems, right? Send a mail, and now though I know it. But if there are any of the doubts, right, keep on sending, whether it is from anatomy, whether it is from physiology, biochemistry, anything, even if we have not discussed, no problems, right? We'll keep one separate session for this, right? Because all those things, they cannot be taken, and it really breaks the continuity, right? So it's like I'm not insulting or I'm not ignoring anyone, but it's like, say, any of the question popping up at this time also disturbs a bit. So please, right, bindas, just send a mail and I'll keep on making the list. Once the list is ready, let's say 10 or 15, no problem, whatever the number it is, whether it is from surgery, gynec, obstetric, pediatrics, anything, no problem. Once that list is ready, I'll post the list in the form of that these will be the topics which we'll be discussing on a specific time, right? In case if it is, because this session will not break. This thing would continue 7:30 to 9:30. This will continue. We'll be taking this session at some other time. So even if you miss it, it will always be there for the recorded part, right? So now onwards, into the even chat also, please don't write anything about the queries part, right? Send a mail and then we'll be having this session, right? And don't worry, it could be from any of the subject, no problem. Okay, done. All right. So thank you for understanding, and we slightly go back. So we were talking about that when this bond is not broken, right? It is what is called as the fiber, right? So it cannot be digested. Uh, okay, okay. Uh, uh, yeah, guys, do you feel, should I turn the chat off? Just for half an hour, so that this important portion it goes smoothly, right? Don't feel bad, right? Just for half an hour, then I'll turn it on. Okay. Should I? Okay. Okay. Let me turn it off. No. So okay. So now this is, chalo, this is good as you wish. Yes, chalo, I'm turning it off for some time. That was a good answer. No, no, Cherry, don't worry. Just for some time, right? So that there is continuity, right? Just for some time. Okay. Thank you, thank you, thank you. Sure. Let's start. So now the chat has been turned off. So we focus fully on our topic, right? Just after half an hour, I'll turn it back again. So bonds which are not broken by any of the enzyme, we call it fibers. And these dietary fibers, because they are not digested, they are not broken. So technically speaking, what would happen? They would add the bulk. They will add the bulk to the stool, right? So this is where like it would avoid that constipation, right? Another thing is that, say, this is the lining, intestinal lining, right? There are fixed number of cells which are there for the glucose absorption, right now we are focusing on glucose. If there are fibers, so these glucose molecules which were getting absorbed Because of fibers, its rate would be decreased. And this is something which is very good because it will avoid the insulin spikes. Some things are like always avoid insulin spikes. Insulin spikes. This is so vital, right? In biochemistry, if we don't remember anything else, it's fine. Just remember this. Avoid insulin spikes. And that's the game whenever you'll be dealing with any of the patient of diabetes or so many even other diseases, right? So it will decrease, this thing would decrease glucose absorption, right? And that is what will avoid these spikes. So it keeps that glucose into its stable condition. When we say that these dietitians, they design the diet, it's nothing but all these principles, if they are taken into consideration, things become much more easier, right? Now we are entering into the second phase. Now in this second phase, we'll be going at a molecular level. So till this point, I don't think there was much of the trouble. In fact, what major thing we learned was just this page, right? So I'm just marking it. It was this page. That is when the sugars, simple sugar and the say complex sugar. In that, these were like monosaccharide, just one, glucose, fructose, galactose, and then disaccharide, sucrose, maltose, and the lactose. This was one page, and this was the second page, that is oligosaccharide and the polysaccharide. In other words, just the chains. Is it a long chain or short chain? Now let's go into the detail of, say, how these sugars would be connected. So starting with a new page with new color, right? Say, now these are the links between those monosaccharides, right? Links. So yes, this is what we said. This is one sugar. This is another sugar. Till this point, we were just drawing it like this. And now we'll add something to it. This is where actually the connection is like this only. But over here it is like OH. Over here it is hydrogen. When they both meet, it leads to formation of water. Water is eliminated and the bond is created. And the bond is created. And this bond is called as the glycosidic bond or the glycosidic bonding. That's what really happens, right? This bond. And then water would be eliminated. So this is link between monosaccharides. On top of it, it is possible that we can add something to it. Sometimes you'll be coming across that alpha 1,4 glycosidic bonding, beta 1,6 glycosidic bonding, right? So whenever it is like, say, alpha or beta, right? When this bond type is starting with alpha, that means this one sugar and this another sugar, they both are in the same line, right? So they are into the same line. When it is like this, this is called as alpha. This is what is called as the staking. So when they are staked over each other, so this is staking, and this is what is called as the linear, right? So that's how the bonds are defined. And when it is said that alpha 1, 4, this means this is 1, this is 4. When it is said beta 1, 6, that means this is 1 position, this is 6 position, right? And the glycosidic bond, right? Same over here. So nothing much. It alpha or beta, it is just telling their orientation, right? We are talking about disaccharide, right? Just to remind, that we are dealing with right now disaccharide, and in between two monosaccharides. So this is the monosaccharide. In between two monosaccharide, we have got the bond, and that is called as the glycosidic bond. That glycosidic bond could be alpha or beta, right? Say if you go for the example, like lactose is beta, right? Lactose is beta. So where it is staked 1 over the, sorry, 1 over the another. Now adding something to it. When the enzymes come into picture, enzymes would be breaking these things, right? Enzymes would be breaking these things. So what they do, say there is amylase, right, which will be acting on large polysaccharides, and then it will be breaking them. But then there are, say, we know about the lactose, right? Lactose was what? Lactose was the formation, was formed with the help of glucose plus galactose, correct? So this lactose, or let me write it here, so that would be much better, right? Say lactose. So then the enzyme lactase, correct? Enzyme is always ending with ase. So when it attacks on this lactose, it has got just two components. So it has to generate glucose and the galactose. Done? So no big rocket science. We just know that they are made up of what? Say for example, that sucrose, right? Sucrose. So enzyme name would be sucrase, right? Sucrase, ASE, sucrase. This sucrase will attack on sucrose and it will be converted to glucose and fructose, right? So because it was made up of that glucose and fructose. And when it comes to say maltose, right? So obviously it would be maltase, right? And glucose plus glucose. So two glucose molecules. The idea is that whatever we do, we have in our final kitty, we have got either glucose, we have got either galactose, or we have got fructose. Bas, that's it. Even if it is a long chain, finally the chop, finally they'll say, okay, we have got these currencies, these three, glucose, galactose, and the fructose. Now you deal with that. We are pouring this thing into blood, and blood, you handle them, right? So from entire GIT, our body is not understanding whether we ate that pizza or we ate that makai ki roti or whatever. It will say just finally this glucose, galactose and fructose in different proportions, they will be coming up, right? They will be absorbed in GIT, right? And from GIT they will go into blood. Now the entire game will be played into the blood, right? What happens? Who is sensitive? No doubt, right? It is insulin. So as soon as this glucose enters into blood, right, we know that it leads to immediate release of insulin. Insulin will say what this glucose is doing, right? Let's do the fixed deposit, right? So it will say send this thing, and liver do the fixed deposit in the form of glycogen, right? So glycogen, till this point we were telling that it is glucose and glycogen, but basically glycogen is what? Glycogen is also a type of sugar. But because now everything is packed, right? Everything is packed. So this glucose was what? Monosaccharide, right? Monosaccharide. This glycogen is polysaccharide. So tuck, all those glucose, they are linked. Now they cannot be broken. So they are all packed and they are stored. It is exactly like that. Sometimes the books which we don't read, and we just pack them and we put at one place, right? So same thing happens in this, and this is what is called as the polysaccharide, right? So that's how this entire system works. Now in this polysaccharide, for that is for this glycogen, it uses alpha 1,4. Now you know alpha 1,4 glycosidic bond, right? Glycosidic bond. It uses even other bonds also. So the point is that now these bonds, they cannot be broken so easily, right? So that's why it is stored. The glucose, it's not so that entire thing is done in the form of FD, right? This glucose, first it will be converted to pyruvate, and now you know it very well that this pyruvate is so important, because it is this pyruvate which is actually in that entire cycle of that glycolysis, right? It is what is called as the glycolysis. It uses oxygen, right? And say this process it occurs invariably, because that's like a link between respiratory system and the digestive system. So once this pyruvate should be reached, and this pyruvate, in case of aerobic situations, right, aerobic situations, when there is enough oxygen, so it will lead to what's called as the citric acid cycle, right, also called as the Krebs cycle. And then there is generation of, that is oxidative phosphorylation, etc. But in short, the energy is released, right? Energy. In case of anaerobic, that's what really happens when we are running, and then muscles are tired, but still we are running, and after that soreness of muscle, right? So that is lactic acid or the lactate, right? Lactic acid or the lactate. And when this is accumulated, that leads to that sore muscles. The idea is, over here, the main player is this glucose. He is going for that entire game, that what exactly is to be done, right? Now it's like glucose is told that, well, there are other players also. So here is what really happens in that liver, right? So that's the liver. Just for the clarity, right? Someone asked a question and he wrote LEVER. Gentlemen, this liver is that liver which is for engineering students, right? So in which there is fulcrum, there is load, right? There is power, right? All those things, right? For us, it is LIVER. Take care. Just telling. Okay. So over here, we do have glucose, right? That we have sorted out. There is galactose, right? There is galactose. Now, when this galactose is there, this galactose will also be finally converted to glucose only, right? It is because it is the glucose which is in use. And how to do that? It is just a game of flip. On fourth carbon, right? This is the location. If you remember it, it's fine. If not, no worries. But just this is the location, that at fourth carbon, it is HO, instead of it becomes OH, right? That is what is called as the flip, right? So instead of HO, now it is OH, right? The bond which is connected like HO, it is now connected like OH. So this will call it a flip, right? Just ulta. And that becomes glucose. And then once it is glucose, so now the entire path is easy, right? It goes like that. When we say fructose, right? This fructose, it is 6 carbon, right? It has got 6 carbon. This is interesting because it breaks, right? You must be remembering that fructose, say, we were telling fructose 1,6 bisphosphate, right? So it was like it was breaking into, say, 3 carbon molecule structure and such 2, they are created, right? Such two are created. And then they will be absorbed all the way. They will be going into the form of glycolysis, and the process, it goes on. So this is what goes into the glycolysis. So whatever it is, whether it is glucose or galactose or fructose, finally they all are dumped into the glycolysis, and whatever we have eaten, finally it will be converted to these monosaccharides, and they are utilized, right? So final production is the energy. Now this will take us to in one important factor. It is what is called as the glycemic index or glycemic index. This glycemic index is a very controversial thing. That's why there is one more thing, what is called as the glycemic load, right? Let's put it into simple words, what exactly glycemic index is. Now you take any of the food, right? Just 50 grams of food, of, of food, 50 grams of food. And then you take 50 grams of glucose. Obviously, this is 100% pure glucose, right? This is 100%. And this 50 grams of food, that would be consisting of variable amount of glucose. When both are consumed, over here, there would be rise in the blood sugar, correct? When this food is consumed, whatever the food it is, right, there would be some amount of glucose into it. So accordingly, there would be some rise in the blood sugar. But so this thing, this rise is what percentage of this 100%, right? That after a specific time, after time, say t, let's take it like say 10 minutes, right? You measure that what is the glucose level right now. Let's say it is 100 and say 50. So this rose how much, right? Everything, let's say, started from 100, and this went to from 100 to 125. So this 125, this is 150. If this is 100%, so this is 50%. So this means its glycemic index is 50%, right? How much rise this is in percentage of the 50 grams of that pure glucose against 50 gram of food, right? Now this is a very ideal condition, because all the foods, right, all the foods, they are having different amount of glucose, true. And another important thing is that why this is, say, when something is having high glycemic index, so it means that it is getting broken very rapidly. It is broken fast, right? It is broken fast. And similarly, if it is low glycemic index, that means it is broken slowly, right? So broken slowly. Example, we just saw fibers, dietary fibers. So dietary fibers will be having low glycemic index, right? Because they'll be going slowly. Even the protein, right? Protein, right? It will be going very slowly. But on top of it, there is one more important thing. That 50 grams to chalo you said is fine, this 50 grams of food, right? But this 50 gram is having how much carbohydrate? Now that is also very important, that how much carbohydrate we have got, because we know that if it is a simple sugar, it is going to spike because it has got nothing to do, straight away it comes, it dumps into the blood. If it is disaccharide, okay, it will break into two. If it is say polysaccharide, so it has to just chop, so yes, it is taking more time, right? But now in this particular food, again, the food is never like, God has never made the food like, okay, only simple sugar, only disaccharide, only polysaccharide. No, it is always a mix, right? So that's the reason that the new concept or the new, say, terminology in the form of what's called as the glycemic load. Glycemic load. The best example is like watermelon, right? Watermelon shoots the glucose, right? But if it is consumed in less quantity, nothing happens. Yes, it is having high glycemic index, right? High glycemic index, so it rises. But the amount of carbohydrate, amount the carbs in say 100 grams of watermelon, it is just 5. It is just 5 grams of carbs. Just 5 grams. Against that, you pick up any of the food packets, right? You see how many carbs are there. So it is like even if we consume that even 10 or 15 those potato chips, right? They will be spikingly high because the quantity of carbohydrate into that much is extremely high. So that's the reason that it is the glycemic load, that how much carb are present into that much food is also a factor which is to be considered, right? As it will be giving some very important clues. And say in when we'll be talking about in medicine, when we'll be talking about management of diabetic patient, right? So in that we'll be talking about again this concept, right? That how it works. Okay, this takes us now to our second point, and this is protein, right? Guys, this is going to break so many myths. These gym people, right, they keep on doing all those false advertisement, right, just to sell those proteins. After learning this protein, you yourself will understand that what exactly is needed so that you remain strong, right? And at the same time, when you'll rotate that dabba, right, and in that you will read that okay, what exactly are the contents of that protein, you'll be able to understand that many of the contents, they are, even if you don't get it from it, they are available, easily available even in your day-to-day diet. Only some of the contents, they are vital. So, and at times they are absent. So there is a big, actually, though it is like live, but yes, there is a scam sort of thing going on. So many all false things, right? And I'll keep on telling you, just I'll not take name of any of the company, but I'll tell you that how they really say advertise those things. So what protein is? When you do exercise, there is always an injury, right? But this injury is called as the micro injury, right? You do any of the exercise, but then protein comes into picture and protein repairs it. After repair, that muscle is much more powerful, and this is the basic principle of any of the, so now it is much more powerful. This is the principle of any of the exercise, right? It is much more powerful. Now when it is, so in this case, when they say, okay, you have finished the exercise and immediately post exercise, this is a post exercise protein drink, right? This is pre exercise, pre workout protein drink, and this is post workout protein drink. And they will try to sell you both the things. It is completely nonsense. Means, is it like after exercise you consume that protein and talk, right, instantly, right, as if it is an electronic circuit, and immediately the entire body, the current has gone and all the muscles are repaired? It never happens like that, right? Today we'll be discussing that it has to pass through so many sequences, and those sequences are so long, it takes time. Some of the peptide bonds are to be broken, then and then the body can and the tissues, they can actually utilize it. The correct method is, say tomorrow morning, if you are planning to run a marathon, right, you have that protein today before 12 hours. Give your body so that it absorbs that properly, and remember not to overload yourself. Now in case even partially, even if you have overloaded yourself, nothing wrong can happen. Nothing wrong can happen partially overload, right? But not extreme overloads. So normally it is like 0.8 gram of protein per kilogram of your body weight, right? Of your body weight. So if you are, say, about 70 kilograms, so you will need about 55 to 60 grams of protein, which is fair enough. This calculation changes. This is for your normal workout. Now, if you are going for heavy workout, right? And when we say heavy, in fact, say I would add one point on the day when we'll be discussing those miscellaneous topics, isometric and isotonic exercise. If you are knowing about it, very well. Else, say it because it will take more time. That's when right now not discussing. But those isometric and isotonic exercise, how can you really combine? I always feel because you are doctors, when you understand these things, you should be super fit, right? You should be super fit. So there we'll discuss, and all these exercises, they will never take more than 20 minutes. Just it is a matter of how well you are planning it. But these 20 minutes, when you are doing, these 20 minutes are mine, right? No mercy, nothing. But then see the performance and see how well you improve. Seriously, not more just after 21 days. Within 21 days, people would say that, boss, what's the reason? What's the secret? Right? That's why it is said that when you consistently do exercise for 1 day, 2 day, 3 days, right, you start feeling the difference. When you consistently do it for 3 weeks, right, people would start feeling the difference. So that's the key. Okay, so coming back to our point, that when we say proteins, so technically proteins, they are nothing but amino acid. Now, if we are introducing a new term, amino acid, that means we need to know that what exactly is this amino acid. That means it must be a chota sa component. If it is amino acid, so we'll just now onwards write it like A. So this is AA attached to another A, attached to another A, and so on. So this bond between two of them will take a new color, and this is what is called as the peptide bond, right? So this is peptide bond. Now these are long chains, right? Very long chains. So then those chains, they keep on twisting, 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 and then how they twist and what is the final output, that is nothing but a protein. So every protein has got a different signature, right? Different signature, what I mean, different twist, different shape. Every protein, right, that has got the final shape. So we'll say that they twist and fold to form new proteins. Now let's go into the detail of our amino acid. It would be better if we write full, right? It makes the meaning of it. Amino and acid. This and this. So it means there are two things which it is forming. In the core, there is carbon, right? There is carbon. With one side, there is amino group, right? So when we say amino, it is NH2, right? This is amino. So here it is NH2. So that means this is like amino group. All right. So it means on the opposite side, there must be something which will be having the word acid. And yes, you are right. It is COOH, right? COOH. And this part, this portion, right? This portion is called as carboxylic acid, right? Carboxylic acid. So one word taken from here, one word taken from here, and the khichdi is made amino acid. So basically amino acid is like on one side amino group, other side carboxylic group, combine both of them and then you get it. But wait, there is something extra, right? And that extra is, there is one hydrogen, right? He is bichara alone. And then there is a stylish chain. This is what is called as the side chain, right? This is a side chain, and this side chain is deciding so many things, right? So in other words, the normal protein would look like this. There is only one exception, right? And that exception is proline. This is proline. Proline is not this complicated, right? It is just a simple ring-like structure. So just to know, it is just like that ring-like structure, a small protein. As such, there are more than 200 proteins, even more than that. But our human body, it picks up only 20 proteins. And out of those 20, it will make almost everything whatsoever is needed for the body, right? So combinations, so they are like hundreds and hundreds. But out of all, right, say 200, 2000 is a bit too high, right? Say 200 plus proteins, right? Human body would use 20. It would use 20. Now these 20 proteins, the next 3 pages, try to write it down 10 times, 15 times, right? You will be very benefited in a long run. Very, right? These 20 proteins, I'm not telling that you just I'm writing 1, 2, 3, 4, 5, the list. But these 20 proteins, let's divide into some parts. The first group is we get it from food, and in case if not, our body is powerful, body can make it. So it's like happily we can make it, we can make it. So if we can make it, we don't have to buy it from anywhere else. So it means we'll call this as non-essential. Non-essential doesn't mean that it is not essential for the body. Non-essential means because we can make it, that's why there is no need to buy from outside, right? We can make it by ourselves. There are 5 such. To remember this, remember it like gas, right? Remember it like gas. This G is glutamic acid. These 3A, right? We'll write it in a different way. The first one is La9, right? La9. They are quite musical, right? And then aspargine, right? So this 9 and gine, they go quite rhythmical. And then this is aspar, this is tic, right? Acid. So it is aspartic, right? I think you must have got it. Aspartic. So instead of this, we just wrote this aspartic. And finally this S. Here also it is ein. So this is also serine or serine, right? So this is not that difficult, right? Just you will write it 2, 3 times and you will be able to remember it. Okay. Its effect would be very interesting. If the question comes, right, from that which type of protein is this, etc. But at that time you will be so confident, and still better is that when you go to any of the stores, you rotate that dabba and you say that, okay, these are the proteins which are already present, right? That is where you start deciphering that what is the amount of protein into that substance. If these glutamic acid, alanine, aspargine, aspartate, serine, if they are given, then what's the point? Why are you spending money on that when it is already present into your food and your body can also make it, right? In many of those protein dabbas, these are the proteins which are added into that because no one knows, right? And they are easily and cheaply available. So fine, right? People and they said, see, there are so many proteins, right? It really happens. It really, really happens. Okay, so this is non-essential. The next one, what is called as the conditionally essential. So we'll write it like, say, conditionally essential. It means what? It means that as such, our body can make it. But if the person is sick, right, when his health condition is not good, at that point he might not be, right? So that's why we say that the normal body can make it, can make it. For this, we'll remember project, right? Project, but we'll temper with the spelling. The project is JECT, no, but we are remembering like GACT. So here it is. So 1, 2, 3, 4, 5, 6. These are the 6. So this is pro is 1G, 2G, A, C, and T. Right? Let's write them down. Proline. Proline, we just talked about it. What was that? That this proline is the only protein which is not having long chain. It is just a ring-like protein, right? So this is ring-like protein, right? A ring-like protein. Then we have got G, 1G. Glutamic acid was already utilized, right? If you see, here it is, glutamic acid, right? We have already utilized that glutamic acid. So then what have you left for me? Well, glutamine, right? So it is glutamine. And then a short one, that is glycine, right? A, C, T. The easiest is tyrosine. And this is arginine. So then why should I write it like this? Arginine, right? And then cysteine or cysteine. Yes, in order not to mix them up with each other, you might have to write it 3-4 times. You might have to just write it and stick it on the wall. So aate jate, just keep on watching, and very soon they'll become part of you. But always write it like this, Arg9, right? Don't write Arginine, no. Right? This gives tremendous visual impact. Right now you'll say that yes, we remember this proline. But when you every time whenever you write proline, if you write proline like this, na, sorry, if you write it like this, proline, or I just got the idea. We can also write like this, right? Because this is what line, so proline. Right? So that way, you'll always remember that it is a ring-like protein. Okay, so 5 plus 6 means 11 down, right? 9 to go, because we said that total there are 20. Okay, now these 9, last 9, they are very vital, and because we really cannot make it, right? So these are 9 which we get only from food. When we are getting it only from food, so we'll call them essential, right? As such, so all are essential, but this is like, because we have to get it from food, that's why it has to be into our grocery list, right? Like say dairy, like those seafood, right? And in case if you are say having non-veg, so then it is like say in meat or say soya beans, right? Best is tofu. Tofu is very good. Tofu. Right? So all these things. So these are 9 essential. There was an actor called Terence Hill, right? And there was a very nice movie, Blackbeard Ghost by Terence Hill and Bud Spencer. Very hilarious movie, right? And then there were 3 parts of it, right? Very interesting, very interesting movie. Bud Spencer was a ghost, and Terence Hill was like a normal guy. He was trying to impress uh, one of the girl who was studying with him into the college. And then there was a basketball match, and it was so hilarious, right? If in case if you get that, just watch that basketball match. Amazing, because Bud Spencer was like bhoot, right? So no one could really see him. So when that those guys, the opposite team, they were those big, bulky, huge guys. And then the moment he was trying to do the basket, right, Bud Spencer would hold him from his back. So then when that person would jump, right, now they were showing it in 2 shots. One shot when Bud Spencer has just hold him from his back, and that person just drops the ball like this instead of jumping all the way and doing like this. And then they show the real shot, right, in which the Bud Spencer is not there, but then how that expression on that person's face, that why I am just dropping the ball like this, right, instead of jumping all the way. Amazing movie, right? So that was like Terence Hill, and there were three such movies. So what if this Terence Hill, right, he is now so famous, he was famous, and Terence Hill met the vice president. So here is the Terence Hill, there were 3 movies, met vice president, right? So from H, right, it is histidine. Now entire sequence is very easy. It is isoleucine, right? Isoleucine. So this next one is leucine, and one must look for these say proteins, right? They are very essential, so they must be there into that protein dabba. Then lysine, right? Lysine. So this hill part is over. This is what I like the most, right? The time is 3:09. Right? So what have we done? The name is 309. Right? That's the name of the protein. So this is 309, right? So that is 309. So this is the name of the protein, 309, right? So then this 9 is something which we like. So that's why it was taken like this. So this would be methionine, right? Again, methionine. This T tyrosine we have already taken. So now this is tryptophan. Tryptophan. And V is valine, right? So how many there are? 1, 2, 3, 4, 5, 6, 7, 8, and 1 is left out, and again it is our 9, right? And because it's the end, so end should always be great. So that's why it is phenyl LA9. Phenylalanine. Done. All these 20 essential proteins, they are done, right? So these essential proteins, these are like dietary proteins. So any protein whatsoever is taken, this protein, right, they will be called as the dietary protein, and they will be broken by the process, right, called as proteolysis. They will be called as the proteolysis. And now starts the journey. Here is the stomach. On next page, we'll go into the duodenum, right? Here is the stomach. So the food which we ate, whatever the food, right, it would be attacked by hydrochloric acid into the stomach, right? We are into the stomach. So I'm not writing, you know that, right? So it would lead to denaturation. It denatures the protein. What is this denatures the protein? Nothing but because this protein was a very complex, those goal, right? This was the protein, right? We just make it simple, open it, right? So now it is like this. So that is what is called as the unfolding. So that protein is now opened up. It is unfolding. Once the protein is unfolded, now it is easy to act on it. True? So there comes the pepsin. Here is the pepsin. And from where it will come? Because this pepsin is going to chop it, right? It is going to break it. So this pepsin is released by those chief cells of stomach. So these pepsin, they will be chopping into those oligopeptide. We know that what is oligo, right? Oligopeptide means few, right? So they will be chopped into the smaller sections, the smaller ones. This much work is done in stomach. So 1, 2, and the chopping part, right? That is done. Now we enter into the duodenum, right? So here is the duodenum, and in duodenum we must know that this is what? This is D2. That is second part of the duodenum. Second part of the duodenum. So we have to draw something. Which organ? Right? Yes, you are right. Though it is not in proportion, but it is okay. Right? Here it is the pancreas. In pancreas, there is a pancreatic duct. That's right. This pancreatic duct will open into ampulla of Vetter, right? So you know this thing very well. So I'll just write this is pancreas. Oh, why to write? You know that, right? You know that. Okay. So now it is into the second part of the duodenum. So there enters those oligopeptides, right? Those oligopeptides, they enter. These oligopeptides, once again, they are fired by this pancreatic exocrine secretions, right? So, and via this ampulla of Vater. So here they are with that gun ready, right? And then there is bombardment, right? Tuck, 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 right? And the final output is they are literally broken into either tri or di or monopeptides. Monopeptides, right? So it is all thanks to this pancreatic exocrine secretion. Good? Exocrine secretion. So they just break it into it. Now once they are broken, they will start traveling. Because this is duodenum. After duodenum, there would be jejunum, right? Jejunum. After jejunum, there would be ileum. So it is a long way. It's a long way, right? All the way. So now what really happens into this path? Just simply one thing, and that is all these are intestinal cells, right? So we add one more word, intestinal absorptive cells, right? Absorptive cells. These intestinal absorptive cells, they are called as enterocytes. Enterocytes. Now what they do? These enterocytes, they will say that once intestine are there, right? They are broken into all chopped into those smaller component, right? So that is all amino acids, right? Di, tri. So some of them, they will remain there only into these enterocytes. They'll say, let's not send them to any of the place, right? They are these intestinal cells. They will say, okay, we are keeping something with us for, we'll use the word for local repair, right? For local repair and local secretion. So they are just kept with them. Rest are all released into the bloodstream, right? They are released into the bloodstream. And then from bloodstream, they'll be moving on to various tissues for that reparative process, right? Whatever, because protein is used at so many places, so it repairs, it helps in so many secretions, right? It helps in all those contractions, right? So many processes they are, thanks to protein. So this is what really happens. And this is the main reason that just before exercise or just after exercise, if any of the protein is consumed, it is not going to make much of the difference. I'm just saving this file. Okay, saved. Now we move on to the third portion, right? This is going to be so interesting. Fats and lipids. Fats and lipids. Again, they are major source of energy, right? And they help in so many other systems for the absorption, for the regular systems. Now, its formation is bit tricky. Bit, not very tricky, but bit tricky. They are made up of two components. One, what is called as the glycerol. Glycerol. Right? And second is fatty acid chain. Right? So there is, and this fatty acid chain, this fatty acid, I think we have heard of it, right? This is the fatty acid which was getting utilized into that gluconeogenesis, right? And something which was non-carbohydrate source of energy, right? In the gluconeogenesis, along with amino acid and fatty acid. This is the thing. So, but they are right now they are bound. They are not separate. So, what this glycerol is, right? So, this is like three, and I'm drawing it because once we'll draw, just within seconds you'll understand so many concepts, right? If you draw, you can even draw it along with me also, right, in your books. So three carbons, they are connected, connect them, then put all the way the hydrogen, and then OH, OH, OH. The best part is that that bonding is always in the same style. So this is what is glycerol, right? This much is glycerol. Pretty easy. Now fatty acid chain name itself is telling that it is chain. So chain of carbons, right? So chain of carbons. And it is a long chain. All the places they are filled up by all those hydrogens, right? Hydrogen, hydrogen, this, all hydrogen. So I'm not writing, right? Except there is a carboxyl group. So O and OH, right? This is the area which we are interested in. This is the area. And this is the area. So OH, they both meet. When they meet, what it is? H2O. H2O leaves. And now we have got a bond, right? We have got a bond, right? So this bond, because now this oxygen is gone, so, and this OH is gone, right? So I'm just drawing it like this, is gone. this is gone. So practically speaking, it is this carbon which is now connected to this carbon. True? This is how it forms. And right, you got it? That OH and the O from both the sides, they are knocked off and it is connected. Now when we have got such, now we'll make it a bit easy, right? So this was C, C, C. On over here, if it is one fatty acid which is connected, one fatty acid chain, right? One fatty acid chain. So what we'll say? We'll say monoglyceride, right? So glycerol was there and it has got one fatty acid chain, so it is monoglyceride. Now this type of fatty acid chain, it can be connected here also, right? See over here. This is also an OH and a new chain is there, a new chain, and it gets connected. So now it has got two fatty acid chain on one glycerol. So this is also a fatty acid chain which is connected. So in that case, here also, so then both of them are, so obviously it will be called as diglyceride. And you must have guessed that what if third one also? Yes, very right. And that also fatty acid chain, when all of them they are connected, so it is called as the triglyceride. And now this rings the bell, right? Triglyceride. So when they say, "Arey, triglycerides are very high," yeah, it is a matter of concern, right? It is a matter of concern. So when they are triglyceride, this is all good. But here, one more thing, that these fatty acids, this fatty acid chain, we to just said that okay, this is a fatty acid chain, but how long it is, right? Well, it depends upon the carbons, right? That's how we saw it over here, the 1 2 3 4 5 6, right? It could be anyone, any number. So this is when we say fatty acid chains, it depends on number of carbons, right? And these number of carbons, if there are just 2 to 5, so obviously we'll call it a short chain, right? If there are just two, so we don't even call it a chain, right? But well, up to 5, right, it is the short chain. 6 to 12, it is like a medium chain. And then comes the long chain when it is more than 13, right? This is long chain. Now I tell you one very interesting thing. There is a very beautiful concept of MCT, medium chain triglycerides. These medium chain triglycerides, they are actually extremely beneficial to the for the health. Now I'm about to write something, right? Try and do it once and you see the effect, right? What you have to do is take a cup of water. To this, this is available in the form of MCT oil. It is called as MCT oil, or you can right now you can add even just to test and the taste, right? And just to test and the taste, add the olive oil. Well, so far though it is okay, but then on top of it you add the coffee. Now it has to be black coffee, right? Add some, say not the sugar, but sugar free, right? Any of the artificial sweetener, aspartame is fine, right? So you can use aspartame. And then once this should be the boiled water, right? To that you add olive oil, and then you add coffee. This is MCT oil also, it is available, right? It is the best, but just for test you can use olive oil. And then put that coffee into it, mix it, and wait for 3 minutes. Let it seep, right? That means simmer. Let it remain as it is, and after that you have it. You'll be surprised at your energy level, right? It will skyrocket. This is what is called as the bulletproof coffee. It is called as the bulletproof coffee. Don't use it every day, right? But let's say if you're planning to go for a heavy workout, right? Try it and see the difference, right? And because you are utilizing those simple basic things, it is non-addicting, nothing will go wrong, but very powerful, right? And your body would like it. Okay, so coming back to our fatty acids, this is another area where these advertisement guys, they really cheat you royally. They cheat you royally. In fatty acids, there are saturated and unsaturated. I used to forget about that which one is good and which one is bad. So I remembered it like that this S means sada hua and U means uttam, right? Uttam means very good, right? And this S is sada hua. So then things fixed up in mind. That yes, it is saturated means that there are all the hydrogen atoms fully loaded. Hydrogen atoms, they are now full, right? There is no space for anyone. And in unsaturated, it means there is space for hydrogen. They are not full. That means, okay, that is about the hydrogen. But what exactly it really does in this? It does it in this way. That saturated means this is carbon, this is carbon, this is carbon, this is carbon, this is carbon. Tuck, 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 tuck. That's how they are connected. This is Sida Sada, simple, directly connected. So obviously when there will be second layer, so they will also be staked like this, right? Staked like this, staked like this. So it means they will be remaining in the same alignment. So that's what is called as the alignment. Whenever there is a good alignment chemically, these things solidify. So they become solid at room temperature. They become solid at room temperature. Now against that, watch for what really happens in case of unsaturated. In unsaturated, rest of the things, so they are okay, but in between these two carbon, they have got the extra attachment with each other, right? Rest of the things, so they are Sida. But these are having a double bond, and this thing can occur even one or more times. It can occur, say, over here also. So it can be at one time or even at more number of times. When such uneven things happen, so it leads to misalignment. There is misalignment. Due to this misalignment, just a minute, let me write properly here. So due to this misalignment, this stake, the way it was staking properly, now it will not go like this, right? So it's like, say, one chain is like this and it has got one extension, another one is having like this, third one is having like this, right? So they are not properly aligned. When things are not properly aligned, it would be into the liquid form. They will not solidify. It will be into liquid form at room temperature. Right? Very crucial component. And if they are at room temperature, when they are liquid, so again, this will give us two things. As we said, that if there is one double bond, right? If there is one double bond, so we'll call it what? Unsaturated. So it was already there, right? Unsaturated. But now we'll call it the monounsaturated. And when there are, say, more number of bonds, right? 2 plus double bonds, right? 2 or 2 plus double bonds. So we'll call it polyunsaturated fats. Simple as that. So whenever it comes like unsaturated, that means they are having double bonds. And if it is mono unsaturated, so that means it is just at one place there is double bond. If it is polyunsaturated, that means at one, or little more than 2 plus the double bonds are, right? Okay. Now when it comes to the digestion of it, right, when it really comes, there is a concept that this double bond is at what place. One thing is very clear, as we said, that these unsaturated, they are uttam, right? They are all good. They are good. They are good, right? And saturated is sadawa. So that is not good. And that's what it leads to all coronary heart disease and so many things. So over here, if it is good, so then we need to know about it. How is it good? It is good for heart. Okay. If it is good for heart, so then we know about omega, right? So let's draw it. Carbon, 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 right? And then let's say double bond and then carbon. That's how it is going. This end, this particular end, one end, it is methyl end. And this end is also called as, its another name is omega end. Omega end. So from this particular end, from this point, right, from this point, how far is the double bond? So here this is 1, this is 2, this is 3, and the bond has come. So we'll call this thing as omega 3. When it is like this, see, 2, 3, 4, 5, and then the double bond. So it is 1, 2, 3, 4, 5, or wait, right? It has to be 6, correct? Here. So 6. So now we'll call it omega 6. And same way, right? All the way, this is 1 and this is 9, right? And now the double bond is there. So we'll call it omega 9. So actually, it is like just from this point, that is from the methyl end, how far is the double bond? So it goes into this pattern only, 3, 6, and 9, and that's what it is called as the omega 3, omega 6, and the omega 9. Now all these omega, they are good, right? They are good. Why they are good? Obviously because they are unsaturated. Why unsaturated? Because they are having that double bond, right? So this omega 3 is, when we say, polyunsaturated. So immediately the moment we say polyunsaturated, that means it has got 2 plus double bonds, correct? Right? 2 plus double bonds. So simple. So here their examples on one page, we'll write only the examples of this. This is ELA, EPA, and DHA, right? Don't ask that why they put such cryptic name, right? They could have given such good names, right? So simple names. But anyway, this is alpha, right? At least that we can write. Linoleic acid, right? Lamborghini type of name. alpha linoleic acid. This is, so someone must have drunk and given this name, right? I don't know, right? It is icosa, as if it is Picasa, right? Icosa pentanoic. So penta, that means five, right? So this is pentagon, so five. And all are oikoic, right? So pentanoic acid. Now we must progress, right? This is five, so the next must be six. So here it is, we just write 6. 6 is what? Hexa. And noic is noic, right? Noic is noic. And acid is acid. So here we should give some dhasu name, right? And that is masala dosa, docosa, right? So it is a docosa hexanoic acid. All right? So this is like, these are the examples. Yeah, these examples, they are very important because this is something which is utilized, especially when there is any infection, right? So these are playing some very major roles, like say in flax seeds, walnuts, right? You get this illa. These are from all those, say, fish, right? Say those sardines and salmons. These are the fishes, right, which really gives this EPA and DHA. And say if we are veg, so then even the tablets are available. But at times it is needed. It is needed. These tablets, they are needed. Okay. Can our body convert it? Yeah, we can. We can. Our body can convert partially, because say we eat lots of flax seeds and walnuts, and then will it make this EPA and DHA? It will make. But that conversion is not very great, right? It is not very great. It converts in some quantity. So in such case, say those flax seeds, etc., you have to keep on eating every day. In any case, it is definitely good, right? Because it keeps you really fit. Moving on to the omega 6. Now omega 6, you know that it would be like 1, 2, 3, 4, 5, 6. So just from that methyl end, it is at the 6th location. Again, it has to be polyunsaturated, right? Polyunsaturated, long way, right? The name here, they are bit lino, linolic acid. Then arachidonic acid. That's 2. So this is what we get from all those types of oils, right? And this is from all the fish, meat, etc. It's same style. Can the body convert? Just if we take oils, yes, body can convert, but again, it is not great. Some portion if it is converted. So this is omega 6. Now to even very good tablets are available, right? Omega 3, 6 and 9 combination, and they can be utilized, right? Okay, moving on to the omega 9. This is, now see, it is monosaturated, right? Now in such cases, these are like oleic acid. So they are unsaturated. Okay, sorry. They are like in almonds and olive, right? So that's how this oleic acid, which is really at the 9th position. right? This is also unsaturated. Now, when we were telling about unsaturated, right? Unsaturated. So let's take it on another page. Unsaturated fatty acids. So why we were telling that they are bad, unsaturated fatty acids? One thing we saw that whenever there is carb, so then there are two functional groups, right? So if these two functional groups, they are both on different side, right? These are what is called as the functional group, right? So when they are on different side, one here, one is here. So they can be either on different side or they can be on the same side of the double bond, right? So should I write it again? Let me write it in, let me write it again. It would be much better, right? Just a minute. So unsaturated, in slightly smaller handwriting, unsaturated fatty acids, so that we include everything on one page, right? So unsaturated fatty acid. So over here, two functional groups. They are on the, everything is with respect to double bond. So over here, there is double bond, right? That's what we saw, those double bonds, and they are on the same side. So in such case, we call this thing as cis bond, right? C-I-S, cis. When they are on the different side, so this is what is called as the trans. And it is this trans because here everything is arranged like this, right? So they again, they can stick very well. That means they will be solid at room temperature. And when they are solid at room temperature, right, this is something which is utilized even in the food industry, So this is even at room temperature, it solidifies. That's the reason that it has got harmful effect. It's got harmful effect. In the cis, they are not balanced, right? Because on one side of the chain, it is, there are two functional components, other side is nothing. So it becomes misaligned, and that's why it would be liquid at room temperature. Liquid at room temperature. So that's what we say, those trans fat, right? The word trans fat, it is bad due to this. Now in food industry, what is happening is that normally it is difficult to do it, but there is what is called as the partial hydrogenation. That is the process which they do. And that's the reason that many of these ready products, right, you'll find that even if it is lying over there, still it is not getting spoiled. We try to make those potato chips at home, right? You make it in the morning, in the afternoon, you won't feel like eating it. In the evening, so it is bus khatam. But these outside things, right? They are so good, so crispy. All this thing. And these partial hydrogenation, they are really bad for heart, right? And say all coronary diseases, etc., they are extremely common due to this. So let's see how now the body is negotiating everything whatsoever we eat and how the process goes, right? The way we saw that how it digests protein, how it digests carbohydrate, right? How carbohydrate was broken down, finally the glucose. Protein was also chop, and finally the amino acids, all those needed. And out of 20, make the khichdi, whatever you want, make it, right? All those like jigsaw puzzles. Over here, things are not that simple, right? Here there will be few processes involved. So again, let's eat, right? We eat butter, right? Why to do conjuicy, right? Butter, sugar, right? All those things, right? They are eaten. Triglycerides, they are eaten lavishly. Because when we consume sugar, sugar will also be converted into the triglycerides, right? By those fat cells. So now starts the journey. Here it is. Now in this, the initial part, what we have consumed, right? That's the stomach. So it comes. Now, as such, this butter, right? This butter is oily. And if we try to clean the oil from our vessels, right? You put whatever the amount of water, but still that chiknahat will always remain there. It is because these things are hydrophobic. They don't like the water. So what everyone tries, right from the saliva, right? Saliva tells, okay, I'll try. Stomach would say, okay, I'll also try, right? Pancreas would say that, yes, even I can also try. All of them, they will prepare what's called as the lipase, right? Come on, let's digest this butter. But there is one small organ who is laughing at all of them, right? And that small organ is gallbladder, right? He is laughing that without me you just can't do anything. But I'll come into picture into the second part of duodenum, because here is the pancreatic duct and that common bile duct that enters into this, and then collectively both of them open into ampulla of Vater. This is D2, right? So that's where the gallbladder enters. Gallbladder dumps the bile salts, right? Bile salts. And these bile salts, they are nothing but they are like soap. That's why this process is also called as the process of saponification. Saponification. So why it's like when your hands are very chikna, right? You just put the soap and instantly everything is so clean. It is because, no big thing, this soap, it has got the tendency to break these large molecules into smaller molecules, and then as soon as it is broken, it gets cleaned very fast. Same thing occurs over here. When this large molecules, they are broken into smaller ones, now the lipase will say, okay, now we are happy, right? Now let's attack. And they will start attacking all of them individually. Previously that was so big, bichare, all those lipase, right? They were unable to do anything. So now it works better. And first thing what it does is, it'll say, let's break them, right? So they are broken. They are broken into free fatty acids and monoglycerides. Monoglycerides. Very simple thing, right? Monoglyceride. But still, say they both love each other so much, right? They'll say, okay, theek hai lipase, you did that, they once again meet, right? Right? Maybe it in next janam. And this time they get a new name and they say, right? Micellus. So basically it happens like this lipase bichara, it breaks both of them. Free fatty acid is split, monoglyceride are split, but once again they both unite. And this time they unite in a very unique way. So we'll draw the outer one blue, because they have understood that, see, this is like a river, and once you are in river you cannot take panga with crocodile, right? So they say that this is all watery, so our outer surface should be hydrophilic. So everyone would say कि अच्छा आओ आओ, no problem, right? Everyone would invite us. But inside core is hydrophobic, right? Hydrophobic. So this was what was in Punarjanam, previous Janam, right? See over here, they were all hydrophobic, right? But now they both are separated, unite again, but this time they have become smart, right? So now this smart piece, he starts moving, right? This all happens into the second part of duodenum. Okay, so from duodenum it moves into the jejunum and ileum, right? So obviously jejunum, ileum, they are what, right? Their cells would say, again same cells, right? Intestinal, absorptive cells, and these absorptive cells, they are called as the enterocyte, right? We learned that, enterocytes. So they both are so happy as a couple, but then they know that there is one villain, and that villain is liver, right? He is once again going to destroy us. So now starts a very smart move. They'll say, let's repack ourselves, right? Let's change. So they are masters of disguises. So they keep on changing their roop, right? I couldn't get another word, so I use that word. So they keep on changing their shakal or roop or whatever we say. So again there is a repacking, right? As they are entering into the intestinal cells, right? Because now they can easily go, true? So they can easily go, they will just diffuse into the intestinal cells because they are hydrophilic. So intestinal cells will say, yes, hydrophilic, come, right? So they'll come, and here they will repack, and they will repack to form one very interesting thing, chylomicron. And here they will take the help of protein, right? So this protein is very dayavan, right? He'll say, okay, chalo, theek hai, let me help. So this chylomicron is formed. So this chylomicron is what? It is lipids plus protein. Lipids plus protein. This chylomicron starts traveling. But over here, there is a big danger. Because if it enters into the hepatic circulation, liver would destroy them, right? And now this chylomicron, its size has become huge. Size is now big. So this is like they entered into enterocytes, but now they can't leave. Now they are too big, right? So they can't cross this epithelial cells, right? They are just too big epithelial cells. So they are trapped. So they figure out that there has to be a way, right? And they start searching. They proceed and they come across that those lymphatic vessels, right? Those lymphatic capillaries. So over here, they will open a map and they'll say, okay, from this lymphatic capillaries, right, which is called as the lacteal, right, where will it take, right? So they open that, okay, we'll be meeting our other friends also, all these lymphatic capillaries. Finally, they will be opening into those lymphatic channels, and then this lymphatic channel will take us to thoracic duct, right? Done. If we go via this path, that way we have bypassed that villain, liver, because liver has got no heart, right? He'll immediately destroy this. So that's why they'll say this thoracic duct is fine, because finally all this nutrition should reach to the tissues. So let's enter into thoracic duct. So it reaches to the thoracic duct. Now where thoracic duct opens, right? To see this thing, right? For the thoracic duct, right? This is like say superior vena cava, right? This is a SVC, superior vena cava, and that's like this, that's like this, right? This is the left side, so it is left brachiocephalic vein, right? And this one would be the right brachiocephalic vein, and then it is the internal jugular and the external jugular, right? They will be entering, correct? So this is internal jugular vein and this is external jugular vein. So they drain, all of them they go, right? And then they go into the heart. Okay. This thoracic duct all the way, right? And it opens here. It opens here. No doubt there are some other locations it can open here also, and in case if you read anatomy further, it will say you'll find that it can open here also, or in some cases it can open even distally going behind the external jugular and can open at this location. So I'll just write like 1 2 3 4, but really doesn't matter. Finally though it is opening into just few inch here or here, right? Finally, to this chylomicrons, right? They are into the circulation. That's what it is needed. And this chylomicron, right? Chylomicron, which is so big, right? But in the heart of that chylomicron, there are all fat soluble vitamins. So A, D, E, K, right? These are all fat soluble vitamins. So that is what it will be carrying. From here, finally, in blood, it would be broken, right? It would be broken into fatty acid, and we know that this fatty acid can be utilized, and then finally those monoglycerides, and these monoglycerides, they will be used by muscle, right, for energy, right, for energy, and in adipose tissue they are stored, right? So that's how the entire cycle works. But there is one more important thing, that chylomicron, once it is broken, right, it shrinks, now it is small, and in any case now it will be coming to that liver, and mercilessly liver will end it, right? It will engulf it. So whosoever has been given the energy, etc., it is done, given, and all good, right? So that's how this entire story of our protein, carbohydrates, and the fat goes on, right? I'm just turning the chat on. Okay, chat is on. So as I said, that we'll be going for a very high speed revision, right? So let's go for, just in 5 minutes we'll revise this entire thing. Okay, so like that movie, right, going back. Well, it is like, say, when the H2O is removed, right? When the H2O is, because it will lead to formation of H2O. When the H2O is removed, so it's like C to C. It is a direct bond. So carbohydrate, protein, lipids, carbohydrates and sugars, right? Simple sugar, complex sugar, one or two. These are in the form of chains. So sugar or the saccharide, monosaccharide, disaccharide, and the few are the like oligosaccharide and the polysaccharide, right? Monosaccharides, they were glucose, fructose, galactose, and then combining glucose and fructose, that is the sucrose. Glucose 2 units, they are making maltose, and the galactose, right, that is leading to the lactose. So complex, this is oligosaccharide and polysaccharide. So oligosaccharide in soybean short chains, right? And these long chains, they are like starch. And in case if these bonds are broken, so then they can be absorbed. And if the bonds are not broken, so these are like fibers, right? Dietary fibers. The dietary fibers, they are not digested, not broken. So they add to the bulk, starch, right, to the stool, prevents constipation, and this GI absorb, right, that is the glucose absorption that is decreased because of the starch, or I'm sorry, because of the those fibers. Link between monosaccharides, right? Over here, sugars, they are connected, and this is what is called as the glycosidic bonding. Say OH and the H, right? When they unite, right, leading to H2O, and H2O would actually be removed. So this is where alpha 1 4 or beta 1 6, whenever it is alpha, alpha means it is into the same line, beta is when there is a staking. Amylase, right? These are the large polysaccharide, lactase, lactose, right? Sucrase, sucrose, maltase, maltose. That's how it really goes, and it breaks into those components. Comes to the blood, right? Increased insulin, and then the glycogen, which will be form of a polysaccharide, right? Yeah, I'll see to that. I'll see to that. That's what I said. So here the glucose to pyruvate, pyruvate will be converted to aerobic, and the anaerobic, right, citric acid goes to Krebs cycle, and the energy lactic acid, lactate, and it gives to those soreness of the muscles. Here it is the liver, right, and this is galactose goes to glucose, fourth carbon, and it is just a flip change. Fructose broken into two 3 carbon molecules, and that is the glycolysis. Finally, this is what we talked about, glycemic index, right? How much is actually used for the 50 gram of food against the 50 gram of glucose. High glycemic and low glycemic. High glycemic means they are broken fast. This is slow, right? And in glycemic load, that is how much carbohydrate it has got, right? So higher the glycemic load, actually it will shoot up more of the glucose. Protein, 0.8 gram per kilogram of body weight, right? That is for what we talked about, the exercise part, how much it is needed. Amino acids, right? They lead to all those peptide bonds. In amino acid, we have got the one amino group, second is the carboxylic group, there is hydrogen, and then there is side chain. All these are typically for the signature of that particular protein. Out of so many proteins, 20 are important, and one which is non-essential where the body can make it. So this is the gas, glutamic alanine aspargine, aspartic acid, and the serine. Conditionally essential, they are like project, where G will be written twice, and that's how, say, proline, glutamine, glycine, arginine, cysteine, and tyrosine. That's what we say. And finally, 9 which are only from food. These are essential. That's why how we remembered Hill 3 met VP and all those proteins, right? So this leads to proteolysis, hydrochloric acid, pepsin, and finally it breaks into oligopeptide. And these oligopeptide, they will be broken to tri, di, and monopeptide, and that is what it would be utilized into the bloodstream for local repair. Something is kept for local repair, rest is sent to tissues. For the fat and lipids, right over here, glycerol and the fatty acid chain, right? I'll just see it in more detail and I'll write it in comments. Okay. So monoglyceride, diglyceride, and the triglyceride, that's what it is formed. How many number of carbons are there? 2 to 5, 6 to 12, or more than 13, short, medium, and long. This is what we talked about, MCT, medium chain triglycerides, right? Fatty acid, these are the saturated ones. So when they are into state aligned, they are solid at room temperature. Unsaturated, when there are the double bonds, one or more, this is misaligned, so that will be liquid form at the temperature, right? One double bond monounsaturated, two double bond polyunsaturated. And how far it is, it depends on, that's how we go for omega 3, 6, and 9. That is from the methyl end, how far it is. So omega 3, right? These are the alpha linoleic acid, icosa pentanoic acid, and docosa hexanoic acid. These are the examples of omega 3. Omega 6 is linoleic and arachidonic acid, and this is oleic acid for omega 9. All these unsaturated, right? There are two functional groups. On the same side it is cis, opposite side it is trans. And trans fat, they are like solid at room temperature and artificially done by partial dehydrogenase by the food industry, right? By the food industry, which is bad for heart. And once it is consumed, here is the butter, saliva, stomach, pancreas, they go for those lipase, they work hard, but when it comes to gallbladder, he breaks it into the smaller components, and lipase now acts happily on it. They both are broken, free fatty acid, monoglyceride, once again reunite, but this time outer shell is hydrophilic, so they get the free pass into the intestinal absorptive cells, enterocytes, and this is called as the micelles, right? Repacking is done again into enterocytes because now the liver is there, we need to take care of it. So protein helps, and there is formation of chylomicron, lipid, which is a lipoprotein. Right? Chylomicron is basically a lipoprotein. So this is what really enters. Now it is size is big, can't leave this epithelial cell, so they enter into lymphatic capillaries, from lymph channels to thoracic duct, and from thoracic duct it drains into the blood, right? Why? Unsaturated fatty acids are harmful even when you told they are, no, unsaturated they are good, saturated are bad, right? Saturated are bad. So over here, right, that's where it drains, it comes into the blood, and this chylomicron, right, it is consisting of ADEK, fat soluble vitamin, in blood it breaks, muscles they use for energy, adipose tissue they store it, and finally when everything done it shrinks, and liver it engulfs it, right? So this was for today, and say tomorrow we'll meet again at the same time, right? Thank you so much, and we meet tomorrow. Bye-bye. And this file is saved and I'm putting it into our folder. I've already created a new folder for 3 underscore biochemistry, so I'll be putting it into that.